In this chapter, we'll be taking a look at some of the tools of the laboratory, so the methods that we're going to use to study microorganisms. This chapter is super important because you're going to be doing your lab work at home. So this chapter will give you an introduction to how some of those techniques work. Microbiologists use several different types of techniques to grow, manipulate, examine, and characterize microorganisms in the laboratory. We can basically break them down into 5 specific steps: the inoculation, the incubation, isolation, inspection, and identification. In inoculation, we're simply collecting a sample. In incubation, we're giving it the right temperature in order to grow. As we isolate it, we're taking specific cultures from that growth and maybe incubating again to create more of that culture. Then we'll inspect it, both macroscopically and microscopically. And then finally, we might be using some identification techniques in order to determine precisely what the bacteria is. These techniques are used by anyone studying microorganisms. We call those people microbiologists. This includes students in the lab, as well as researchers that are isolating drug-producing bacteria, for example, from soil, or clinical microbiologists that are determining the source of a particular infection by analyzing fecal samples, by taking a closer look at inoculation and incubation. Inoculation is the procedure that we use to produce a culture. It's adding a small sample, or the inoculum, to a container of medium. Now that container could be either solid, liquid, or maybe it's even a live organism, such as an animal egg. In order to inoculate, the instrument must be sterile. The samples could come from soil, water, sewage, air, many different foods, or bodily fluids or discharges, like urine or feces. After we've inoculated our media, the substance on which the sample will grow, it's time to incubate. During incubation, the sample is kept between 20 and 40 degrees C. After a period of time, there's an observable growth on the medium. The neat thing about having a real incubator to incubate your samples is that you can control the atmospheric gases and the temperature. Once the cultures begin to grow, we can visually recognize the growth as cloudiness in a liquid media, or colonies, literally clumps of piled up cells on that solid media. The growth of cultures is sort of analogous to gardening. We're seeding tiny little pots with cultures. Then we'll collect the cultures, or the plants we're interested in, off that one media and grow them again on another media. In this way, we can exclude contaminants or other cultures that we're not interested in observing. The incubation periods can last from a day to several weeks. You know you've got growth when you have an observable macroscopic colony, such as you see in the plates down here. Now, this is a mixed culture because there are 2 or more species that we can see on it clearly just by the color of their growth. If we were to take one of these and create a subculture, for instance, take an inoculating loop full of the red stuff and place it onto another petri dish full of a growth medium, then we could probably grow a pure culture. Another word for pure culture is axenic. That means raised in a germ-free environment. Now, on occasion, a culture can become contaminated. That is, it'll include unwanted microorganisms or organisms of uncertain identity. These contaminants are like the weeds in our theoretical garden. In this case, we might take a subculture again, removing one of the samples and replating it. A whole new meaning for subculture. It's not just the underground. Now let's take a look at isolation in more detail. When we isolate, we're going to separate one species from another. We separate a single bacterial cell from other cells and provide it a space on the nutrient-rich surface. This will allow the cell to grow into a mound of cells, or a colony. If this colony is formed from a single cell, the colony contains just cells from that species. However, often a colony could contain multiple species. There are several different methods that we'll use for isolation of organisms of interest. First of all, we'll take a look at how we might isolate in general. We have a mixed culture that we've smeared on our petri dish. So the petri dish down here is full of agar, or a growth medium. We have a culture of mixed bacteria, As we smear these mixed bacteria across the petri dish, we'll allow them to spread out and hopefully grow piles of cells of a single type. The first specific method we'll look at is a streak plate method. In this streak plate method, we'll take a droplet of culture or sample that we spread over the surface with an inoculating loop. So the agar, or the growth medium in this plate, has a fairly firm surface. The inoculating loop is used to deposit some of the sample onto the dish. And then that loop needs to be sterilized, and we will literally streak back and forth in order to create a pattern this way. The inoculating loop is then sterilized again, and we'll make another pass, spreading the bacteria further. Again, we sterilize the inoculating loop and create another series of streaks, bringing a sample of the second streak into it to spread it even thinner. And then finally, we'll sterilize the loop one more time and create a fourth direction of streaks. This gradually thins out the sample and separates the cells spatially over several sections of the plate. The key here is you've got to sterilize the loop after each pass, otherwise you could get weeds in the garden or contamination. This streak plate method is the method that's used for most applications in microbiology. However, there are a couple more. Let's look at the loop dilation method. It's also known as the pour plate method because we're literally pouring it into a plate. The samples are inoculated into a series of cool liquid agar medium tubes. So these tubes have been placed in a hot water bath in order to melt the agar. And then it's cooled so it's still slightly liquid. We've put a sample into one tube and then taken a sample from that tube and diluted it further into the second tube. Then we take a sample from that tube, dilute it further, and drop it into the third tube. Usually by the second or third plates, we've created enough space, or less cells per volume, allowing space between each of those cells to allow for colony growth. The key here is each colony has to have enough nutrient on this medium in order to survive. After we've achieved an appropriate dilution, each of these tubes are literally poured into the plate. As they cool, they'll solidify. Now, the only disadvantage here is that some of the colonies are going to develop deep in the medium, so they're not accessible to pick in order to replate and culture again, creating a subculture. Another method is the spread plate method. The spread plate method involves taking a small volume of liquid. It's a diluted sample and it's pipetted onto the surface of an agarose plate, so a growth medium in a petri dish. This sample is then spread around evenly using a sterile spreading tool. The sterile spreading tool can sometimes be called a hockey stick. Again, this allows the cells to be spread out on the surface, allowing space and nutrition for new colonies to grow. So this technique has a similar result to the streak plate method, except that it does require a small volume of liquid sample. Okay, so far we've had 3 of the 5 I's. We've had inoculation, incubation, and isolation. Now we've got 2 left, inspection and identification. In inspection, we use the appearance as well as the metabolism. So we use some biochemical tests and sometimes genetic analysis or immunologic testing to identify organisms specifically in a culture. Now, we can do some of that macroscopically by simply observing the colors of the cultures and the shapes of the cultures. And then the microscope can help us visualize the specific shapes, either round or rod-shaped, or whether they're clumped or separate. This is a phase we call inspection. Beyond that, we need to use these biochemical tests and genetic analysis or immunologic testing to truly identify the organisms that are in our culture. That's the identification phase. Once cultures are no longer being used, they have to be sterilized and destroyed properly. In a lab situation, we would use either an autoclave, which is steam sterilization, or incineration, which is burning of it. Mostly, though, for the purposes of our labs, we'll be using bleach and alcohol sterilization, and on occasion incineration or burning of, say, an inoculation loop to sterilize it before creating a culture plate. Let's have a quick concept check. What term is used to describe a pile of microbes that's visible to the naked eye? This one's pretty simple. We've been covering that one. Colony is the correct answer. Development of the techniques that I just reviewed allowed for growth of microbes outside of their natural habitat. This meant that we could now examine the genetics, morphology, and physiology of various microorganisms. However, successful cultivation had some requirements. First of all, a sterile medium to grow on, and second, all the nutrients that microorganism requires. Now, because of the diversity of microbe requirements, we have a whole lot of different types of sterile media on which all these microorganisms grow. In fact, there are at least 500 different types. These different types can be contained in test tubes, flasks, or petri dishes. We're going to classify all the different types of media, first by physical state, second by the chemical compositions, and then third by functional types. This table below will help you think about this. In the physical state, we consider liquid, semi-solid, solid that can become a liquid, or solid that cannot be liquefied. So that's the physical state. The chemical composition, they can either be synthetic or non-synthetic. And then there can be functional types, which are enriched or selective, differential. Some can favor anaerobic growth, and others can favor transport. Let's begin by looking at media in the physical state. Again, we have liquid, semi-solid, or solid media. The liquid media are water-based solutions. They are literally solutes that are dissolved in distilled water. They don't solidify at temperatures above freezing, and they're considered things like broths, milks, or infusion. When we see growth in them, they appear cloudy, or they have particulates, and sometimes they have a little scum on the top. When we consider semi-solid media, they have a clot-like consistency at room temperature, so they're not really solid and they're not really liquid. Most of them contain some amount of gelatin or agar in them. Because of the thickness, they can be used to determine motility and also to localize reactions at a specific site. The third type of physical state is solid media. It's a firm surface on which cells can form discrete colonies, such as we saw in the petri dishes of agar above. Some of this solid media can be liquefiable and some non-liquefiable. This type of media is useful for isolating and culturing bacteria and fungi. Now, agar is the most common solid media. It's a liquefiable media, so it's solid at room temperature and it melts at boiling point. Once it's cooled, it resolidifies. Agar is made from a specific species of red algae, and it contains a complex polysaccharide that forms this gel-like structure. Now, some non-liquefiable media are things like rice grain or meat or egg. All of these media remain solid both before and after heat sterilization. Next, let's look at classification of media by their chemical content. There are basically two classes here. We can have defined or synthetic media. These media have compositions that are precisely chemically defined, so much so that they have an exact chemical formula. We can see these over here in table 3.2a. Very specific amounts of each of these known ingredients. Now, if we are not aware of the composition of one component, it's not chemically definable, and thus we'll call it non-synthetic media or complex media. In complex media or non-synthetic media, we have a much less rigorous formulation. You can see in table 3.2b the type of recipe that we see for a complex or non-synthetic media. So again, defined media, we've defined exactly what's in there. Whereas complex or non-synthetic, we have more ambiguous things like ground cells and tissues. It might be things like blood serum or meat extracts or milk, maybe even yeast extracts, soybean digests, or peptone. And the final way we classified was by function. We can either have general purpose media, enriched media, selective or differential media, and then a few others we'll cover at the end. A general purpose media is used to grow as broad of a spectrum of microbes as possible. It's usually non-synthetic. They contain a mixture of nutrients that support a wide variety of microbial growth. Examples of this are nutrient agar and broth, brain and heart infusions, or TSA, which is tryptic soy agar. We'll take a closer look at some of these shortly. Another type of function is enriched media. These media are going to support the growth of particular fastidious bacteria. That is, bacteria that require special nutritional or environmental conditions. They contain complex organic substances or growth factors that support the growth of these, let's say, picky bacteria. Blood agar and Thayer-Martin medium or chocolate agar are examples of these. Blood agar is created by using regular agar, in addition adding sterile sheep, horse, or rabbit blood. And then the Thayer-Martin or chocolate agar is created by heating a simple blood agar. In fact, Thayer-Martin medium is used to grow the gonorrhea pathogen. Next, we have selective or differential media, again, by their functional type. Selective media contain one or more agents that inhibit the growth of certain microbes, but not others. We'll take a look at a couple of examples on the next page. Then differential media allow multiple types of microorganisms to grow, but display visible differences between these microorganisms. So it allows us to differentiate between particular microorganisms. Let's take a look. With selective and differential medias, we have two tables here. The one here on the right includes differential medias, and down here below we'll see selective media. Here are a couple of examples. When we consider mannitol salt agar, in this plate here we've used mannitol salt agar. Mannitol salt agar has a very high percentage of sodium chloride. It's about 7.5%. This is a selective growth media for Staphylococcus bacteria because they can tolerate this high salt concentration. Also, it has the sugar mannitol in it. So those bacteria that can digest mannitol produce an acid which turns the red indicator you see in here bright yellow. So mannitol salt agar is both selective because it allows the growth of only things that can tolerate the high sodium chloride. And it's differential because different bacteria that can handle that salinity produce a different effect. Now, here's another example. McConkey agar. This agar selects against gram-positive bacteria. Now, we'll learn what those are later on. So it's selective because it selects against the gram-positive bacteria. But it's also differential because it allows us to differentiate between bacteria that ferment lactose and those that do not. Because as we can see in these cultures, those bacteria that ferment lactose are producing the pink spots. Here's another great example when we look at chrome agar. Chrome agar is designed for culturing the most common urinary pathogens. It's a differential media. You can see in this example, cultures of specific bacteria have been streaked to spell their own names. Now, we're still under the functional categories. There's a few other miscellaneous ones here. We have reducing medias. Reducing medias absorb oxygen or slow its penetration in the medium. These are used for growing anaerobes or for determining oxygen requirements. There's also carbohydrate fermentation media. This media contains sugars that can be fermented, as well as a pH indicator that's useful for identification of microorganisms. If the microorganisms are present that ferment carbohydrates, then we'll see this pH indicator change color. We'll also see that there are assay media. assay media are used to test the effectiveness of antibiotics, disinfectants, or antiseptics. So we'll plate the particular bacteria of interest on that media, and it may contain an antibiotic against that particular strain of microorganism, in which case any bacteria that are susceptible for that antibiotic will not be able to grow. So that's a whole lot of different types of media that we've considered. Remember, there are well over 500 types, so it's better for us to just categorize them in these 3 different ways. We looked at the physical state, chemical composition, and functional type. You can use this table 3.1 as a nice guide to help you think about the different types of media available. Let's take a quick concept check. Let's say X-gal is present in a media, which is a nutrient. Lactose fermenting colonies turn blue, while those that cannot ferment lactose remain white. What kind of media is nutrient agar when X-gal is added? So the key here is we either have blue or white. Is it selective? I'm going to say no, because we can have lactose fermenting colonies or not lactose fermenting colonies. So we'll cross that one off the list. Is it differential? It could be, because we have a differential response depending on whether the bacteria ferment lactose or do not ferment lactose. How about enriched? Well, we've added X-gal, so enriched also fits. General purpose? No, we've made it pretty specific, so we'll cross that one off. So I'd say in this case, either B or C are correct answers. This brings us to the end of our section on culturing microorganisms and the 5 I's. Please review the 5 I's. Make sure you have a clear understanding of each of the processes involved.